says which amino acid to get. And how do we know which amino acid comes in? If it's the correct one, well, it has to have the correct anticodon to bond in there, to pair. And once that happens, once it makes contact, for lack of a better word, with the messenger RNA, hopefully, the correct amino acid will be attached to that. So this is just a molecule transfer RNA isolated. There's our anticodon, which is with its 3 nucleotides. There's the amino acid attached. So here we are bringing in. Now there's the ribosome. Here's the transfer RNA coming in. More transfer RNA molecules coming in. Each one will read a different codon. And it will bring in the appropriate amino acid based upon whatever that codon is. There's our growing polypeptide, chain of amino acids. 10,000 images to look at. You can't hear it once and say, got it. Maybe you can. Most people can't. I certainly couldn't. First time I heard this. You got to work at it. You got to think about it. So one other thing to keep in mind, and this is not something you have to write down, but each messenger RNA molecule is going to be read a lot of times. So once it's read once, and the polypeptide is assembled, it will be read again and again and again. That makes sense. That's efficiency. You don't want to build this messenger RNA molecule and have it read once to make one protein and then have it disassemble and then have to do it all over again. That's not very efficient. So if you have to make a lot of proteins, let's say hemoglobin, we know how many molecules a cell of hemoglobin requires. It makes sense that this is going to be read thousands and thousands and thousands of times. Once it is not needed anymore, what do you think happens to the nucleotides? Are they destroyed, or do you think they are recycled and reused? Recycled and reused, yeah. That makes sense. Okay, how fast does it happen? Again, don't worry about this. It's just kind of an aside. You say, well, how quickly does this, all this take place? Pretty quickly. 20 amino acids linked in a second, perhaps. It's pretty quick. Well, it has to happen quick, right? It can't be a slow process. If you think about the number of proteins cells have to churn out, it has to happen pretty quickly. So there's sort of the summary of what we've seen so far. Make our copy of the gene. There's our molecule with messenger RNA that has been synthesized out into the cytoplasm, associates with the ribosome, and ribosome in conjunction with transfer RNA will assemble that polypeptide. Now, is our protein done yet? No. What do we have? What structure of the protein do we have completed? The primary structure. We have the number. We have the correct arrangement of amino acids. But protein isn't done yet, but might as well be. Because all we have to do now is fold it and modify it a little bit. More video clips.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has 2 binding sites for transfer RNA, the P and A sites.
Again, a little more detail than what we talked about, P site, the A site. Don't worry about that, get the big picture here.
Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG.
So that tells
Transfer
you where
RNA
to
with the
attach.
anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an NI codon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it.
Remember, all this is regulated by the codon and the correct anticodon on the transfer RNA molecule. That spells out which transfer RNA molecule is going to come in and which amino acid is bringing it, is transferring.
The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Okay, it's always helpful to see it. So nice schematic. Keep in mind, most of the images you see on these PowerPoints are right off the internet. Okay, so they're out there. You've seen nice schematic of the whole process here. There's our gene being copied. There's our messenger RNA molecule has been synthesized. Messenger RNA leaving the nucleus, associating with the ribosome. Transfer RNA molecules here, each with their specific amino acid. Anticodon, codon, growing polypeptide there. And then in the cytoplasm, of course, we said we have these amino acids just hanging out there. Where do those amino acids come from? Where'd that pool of amino acids come from? What? Where'd it come from? It's in the cytoplasm. Where'd it come from? What? Where do we get our amino acids?
From broken down proteins that we've
From broken
taken
down
in.
proteins that we've consumed. On that chicken breast you had 2 days ago. And all the amino acids in that muscle protein that have been broken down, and now they make their way into your cells, so you can refashion them. You've taken the chicken, the amino acids that were part of the chicken, now you're making your own protein. And we said some of them, of course, your body manufactures also. Essential to non-essential. Remember that? Sure you do. Let's look at it again.
The inner workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left.
This is your polymerase. That's the enzyme that causes the strand, the hydrogen bonds in the DNA strand to come apart.
Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands.
All these little pieces of yellow, those are supposed to be the what? It's supposed to depict what? The nucleotides that are hanging out in the nucleus waiting to be assembled. into a molecule of messenger RNA.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein.
What are these things?
The
The
tRNA.
tRNA molecules, good, tRNA molecules.
Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
Yeah, go figure that one out, 100 trillion per second. Wow. Everybody's, God, I wish I had something like that when I had intro bio. I had to sit there and look at a book, try to figure this stuff out. So use the internet, such a great tool. It is such a great tool. It can help. Questions? Okay, so we're not quite done yet, right? A little bit more cleanup here. So the next destination of our polypeptide, it will be passed, now starting to fold on itself already. Once you get those amino acids laid down, it's going to start folding on itself. We'll take it to the inner fold of the rough ER. Remember, a lot of these were manufactured right here on the ribosomes, attached to the rough ER, for modifications. So we're going to see protein modification here, but keep in mind the RAR will modify other types of proteins, or excuse me, other types of molecules, like carbs, like some lipids. So this is your aunt saying, wow, I've got the dress pretty much done here, but there are a few things I can't do, and I know a girl, I know a woman, and she's good at, attaching buttons. So we're going to send it over to her we're going to transfer this over to her she lives in Springfield. So the yet unfinished protein, the yet unfinished wedding dress goes to this person, and she modifies it a little bit, makes a few changes, puts on the buttons. so on and so forth. Where to next? What happens? Hmm? No. It'll be packaged into a vesicle. Remember what a vesicle is, a little sac of membrane that pinches off the rough ER. So this woman in Springfield says, well, I've done about all I can, but we need a few last modifications. I know a woman, and she lives in New Haven, Connecticut. So we're going to ship this dress down. We're going to ship this dress down. We're going to package it, ship this dress down to New Haven, where this person can make the final touches on the dress. So where is this vesicle going? Crickets. What? Come on.
Out of the nucleus?
No.
Or out of the
Where does it have to
cell?
go? Where these vesicles that pinch off from the rough ER, where do they go next?
To the
They
Golgi.
go to Golgi, yeah. Sort, modify, package, right? Sort, modify, package. So our next stop is going to be Golgi. There's our receiving side of the organelle, remember that? And then these vesicles dump their contents into the membrane of the Golgi where they are modified, sorted. So this is the woman down in New Haven. She's making the final touches on the dress. And then what is she going to do with it? She's going to package it. So what we're going to see here with these modified molecules, with, in this case, the proteins, usually not talking about just one protein per vesicle, proteins, they will be packaged in a transport vesicle and travel where? Hmm?
Back to the owner.
No? What?
To the back.
Ooh, there's some work to do. It's going to be a long weekend for some of you. Come on, where does it go next? To the membrane, usually. Now, some of those vesicles may stay within the cell, be used. Molecules used there. Some may become lysosomes, as we said, the cellular recycle centers. But most, the contents are packaged and then travel to the membrane where they will be dumped out of the cell, delivered somewhere else in the organism, another destination, where they're needed. So the woman in New Haven is going to package this up. It's going to be shipped to the destination of your wedding, wherever that might be. So the lysosome may develop from some of those proteins that were manufactured. They're so-called digestive enzymes. They may be exported. Or those proteins just may stay right in the cell where they might be needed, could be needed. So a couple different end results here at the end of all this, we can have a couple of different end results. So some of these vesicles that pinch off from the Golgi may remain as lysosomes, circulate through the cell, look for cellular debris, waste to break down. Some of these vesicles that pinch off from the Golgi may just stay within the cell. The contents will be used there. Some will fuse with the membrane and pass the contents out of the cell. Just depends. Just depends on what the particular type of protein is. So we'll say it's exported out, and there is the finished wedding dress to your destination wedding. It's all right there in your book, 168 to 181, if you haven't read so already. It's a lot more detail than what we discussed. in the book, so don't get bogged down in the details. If we discussed it in class, as part of this lecture, pay attention to it. If there's more detail there than what we discussed, you can discount that extra detail. Get the big picture here. Could you describe the process in a big picture kind of way? This is what happens here. This is what happens here. Ask yourself that. Do not walk in next Monday without being able to do that.
Ah, life, a sunny day, a simple friend, and a complex biological story. The Human Genome Project is a way of exploring our molecular selves. Almost all of our cells, the muscle cells that let us smile, the brain cells that perceive the humor in things, the cells of our eyes that take it all in, contain a complete set of all our genes, the genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. A DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life. A simple friendship, a sunny day, a life.
That video was produced a few years back as part of what was called the Human Genome Project, Monster Project. A number of countries were involved with it, and the idea was to read all the human genes, figure out where they are, and hopefully, ultimately, figure out what they do. That hasn't been accomplished yet, but we know about where they lie on the strands of DNA, what genes are where. But the big question is, what do all these genes do? What do they control? What proteins are they responsible for? That's the bigger question. But the first step is figuring out where those genes are, hence the name the Human Genome Project. Okay, there's more. We're not done with this yet, but you have plenty to start working on. So work with somebody. That's a great way to prepare for this. Sit down and explain this process to them. Get comfortable with it. That's a great way to prepare. Remember, none of this nucleic acid stuff will be on Friday's quiz. Friday's quiz is all cells. Okay? Questions? Okay. Have a good morning, everyone.